This is Unit 6 Notes, Cell Growth and Division, Part 1. So we're talking about cell growth and division, which includes the division of the nucleus called mitosis. There are a number of reasons that cells divide. One reason some cells divide is for reproduction. Single-celled organisms like bacteria and amoeba and other single-celled organisms reproduce this way. The only way they increase their numbers is by cell division. Another reason cells divide is because of growth. In order to for a multicellular organism to increase in size, it has to have more cells. You began life as one cell, and now you have trillions of cells. The way you grew is by that cell dividing and producing new cells. Another reason cells need to divide is for repair or healing. When you have an injury, you need to replace the cells that are damaged, and that happens by means of cell division. And a final reason that cells need to divide is because of cell size. When the cell starts to get too big, when the surface area to volume ratio gets too small, it requires the cell to divide in order to meet its needs. Another reason in cell size is that because of the increased volume or decreased surface area to volume ratio, the DNA is no longer to keep up with the cell's needs. So that means DNA overload, which means the cell needs to divide. So what does cell size have to do with cells dividing? Well, when cells increase in size, their volume increases faster than their surface area. So here's the problem. All materials interleave the cell through what structure? Well, obviously through the cell membrane. So the question is, as the cell is growing, can enough oxygen and food get in to meet the needs of the cell? Another problem is there enough DNA to support cell activities? When the cell volume gets too large, the DNA can't keep up with or make enough molecules or enzymes or whatever is needed to maintain the homeostasis in the cell. When this happens, then it creates a critical problem for the cell, and the cell needs to divide. So here's what we're going to do just to illustrate it. Calculate the surface area and volume of 3 different cells and figure out the ratio. And the reason this is important is because the volume increases at a faster rate than the surface area does. The surface area is the cell membrane. The volume is the contents of the cell, the cytoplasm and all of the organelles. All right, so here we have 3 different cells. This one is 1 centimeter on a side. Notice they're shown as a cube for simplicity's sake. We have one that's 2 centimeters on a side and one that's 3 centimeters on a side. Of course, these are much larger than cells actually would be, but we're using these for example. So for the surface area of the 1 centimeter cube cell, surface area is length times width times 6 because there's 6 faces on the cube. So in that case, the surface area is 6 square centimeters. The volume of that cube is calculated length times width times height. So the length and the width and the height are all 1 centimeter, then it's going to be 1 cubic centimeter, which gives us a ratio of surface area to volume of 6 to 1. For the 2 centimeter cube, we have 2 times 2 is 4 times 6, which gives us 24 square centimeters. The volume, 2 times 2 times 2, is 8 cubic centimeters, which gives us a ratio of 3 to 1 for the surface area to volume. And as we increase the size 1 more centimeter yet again, 3 times 3 times 6 is 54 centimeters squared. And the volume is 3 times 3 times 3, which is 27 cubic centimeters, giving us a ratio of 2 to 1. So you can see it doesn't take very long at all for the ratio of surface area to volume to decrease to an extent where it's going to be difficult for the cell, for the membrane and the nucleus to meet the needs of the cell. Now, we call the cell cycle the normal cell cycle. That's the life of the cell from division to cell division. When the cells going about their daily activities in between cell divisions is called interphase. That's between cell divisions. In the cell cycle, there are 4 phases. The gap1 or G1 phase, the synthesis phase or S phase, the gap2 or G2 phase, and the mitosis phase. These first 3, the G1, the S, and the G2, all 3 of those together make up interphase. That's what interphase is. It's the period in between cell divisions. The mitosis, or the M phase of the cell division, is division of the cell. Here's a picture to show you kind of the amount of time that is spent in the cell's lifetime in these phases of the cell cycle. We have the G1 phase, which takes up probably close to half of the cell cycle, depending on the cell. Some cells spend an awful lot more time than this. For instance, your nerve cells spend literally years in this G1 phase because they don't divide very often. Others spend less time, just depending on the cell and what the cell's job is. In the synthesis phase, we have a pretty big amount of time, less than the G1, but more than the G2. The G2 is even less than that, and then the mitosis and cytokinesis in the M phase takes even less time. At the end of cytokinesis, then you end up with two identical cells, each of which will enter a new cell cycle and start the whole thing over again. So here's what happens in each phase. In the G1 phase, the cell is increasing in size, it's busy synthesizing proteins and organelles. Whatever the cell needs to do is being done during the G1 phase. The amount of time spent in the G1 phase varies depending on the cell and what the cell's job is. At some point near the end of the G1 phase, that surface area to volume ratio hits a critical point that flips a switch to turn on the S phase. In the S phase, the chromosomes get copied or replicated. It's necessary for the chromosomes to be copied or replicated so that each daughter cell will have a complete set of chromosomes. That way, the daughter cells can be identical to the parent cell, the original cell, and have all the information they need to carry on the life of the cell. In the G2 phase, in this phase, the production of organelles and molecules that are needed for cell division are being taken care of. Once the DNA is copied, then everything else that's needed for cell division needs to be prepared. So we need more mitochondria for energy. We need the various enzymes and molecules that are needed for cell division to occur. It takes a while for all this to happen, but it all gets copied and prepared before the M phase begins. The final phase, the M phase, is mitosis. Mitosis is division of the nuclear contents. The nucleus has to divide so that each daughter cell gets a complete nucleus with all of the chromosomes. Immediately following the M phase is cytokinesis. This is not really a phase, but this is when the cytoplasm divides to produce two brand new cells. Now, to draw the cell cycle on your paper, okay, you need to draw a circle on your paper. The circle should be maybe about 5 or 6 inches in diameter. Once you draw a circle on your paper and find the center of your circle, then you need to draw a radius from the center to the edge of the circle. Then you need to calculate the percentage of time spent in each phase of the cell cycle. We're gonna have a hypothetical cell that has a 24-hour cell cycle. This cell that we're drawing has a G1 phase that lasts about 10 hours, an S phase that lasts about 7 hours, a G2 phase that lasts about 5 hours, and an M phase that lasts about 2 hours. And if you add all that up, it adds to 24. Now, to figure this out, you have to calculate the number of degrees each phase requires out of your circle, and then divide your circle to appropriate segments. For instance, the G1 phase lasts 10 hours. If you divide, if 24 hours is equal to 360 degrees, 1 hour is equal to 15 degrees. So the G1 phase lasts 10 hours. 10 times 15 is 150 degrees. The S phase lasts 7 hours. 7 times 15 is 105 degrees. The G2 lasts 5 hours, which gives us 75 degrees, which leaves 30 degrees for the M phase, which is 2 hours. So what you're going to do is you have to draw a radius, put the center of your protractor on the center of your circle and line it up with the radius and measure off 150 degrees and make a mark, draw a line from the center to that mark. Rotate your protractor so that now the new line that you just drew is on the 0, and then you need to measure 105 degrees from that point and draw another line. Rotate your protractor again and measure off 75 degrees, draw another line, and that should leave 30 degrees in your last section. If you would color code each segment, then and describe the events of each one in the diagram. Leave the M phase blank. We'll fill it in later. This is what your diagram should look like, something similar to this. This shows a 24-hour day with the segments for the G1, S, G2, and M phases of the cell cycle. This concludes the notes for the first part about the cell cycle.